Greetings again, and welcome to the second part of today's lecture, which is going to cover the detailed structure of the skeletal muscle. So, the basic structural units found in skeletal muscles are called sarcomeres. Now, just so that we're clear, sarcomeres are not skeletal muscle cells, okay? In order for a skeletal muscle cell to be formed, it needs to have multiple sarcomeres. So sarcomeres make up skeletal muscle cells, and then skeletal muscle cells will join together to form one large muscle. So multiple sarcomeres are joined together in order to form one skeletal muscle cell. So what are sarcomeres in any case? Sarcomeres are basically sorry, I just had a technical problem with my charger. So sarcomeres are, like we said, the structures that make up skeletal muscle cells, and they are made of what we call protein filaments. So if you look at this picture, you can see that you have, uh, sorry, lengths of, let's just call them threads, okay, that are arranged parallel to each other, okay, and then they're kind of arranged in like a let's call it like a step-wise parallel manner. So you've got alternating between this pink filament and this blue one here, and they're just kind of stacked one on top of the other. Okay, so these protein filaments and the way that they're arranged make up what we call a sarcomere. So sarcomeres are made of thread-like proteins that are called filaments, and filaments are arranged longitudinally. I realize in the picture it was more of a horizontal arrangement, but they're arranged along the length of the muscle. And they run parallel to each other such that they are able to slide on one another. So basically just think of it kind of like um kind of like a machine, okay, where you've got wires that are kind of working together and moving on top of each other, okay, so one of them slides and then the other one pulls it and then they end up sliding together, and this is what ends up causing movement and change in muscle structure and contraction and so forth. So the proteins that make up sarcomeres, so we've agreed that sarcomeres are made up of proteins. And sarcomere proteins are two types. They can either be filaments or what we call accessory or regulatory proteins. So basically the main structure of the sarcomere is made up of filaments, but in order for these filaments to function, in order for them to actually slide on top of each other and cause coordinated movements, they need to have certain proteins that help them with this function, and these are what we call regulatory or accessory proteins. Now, the filaments themselves can either be what we call actin filaments or myosin filaments. So the overall structure of sarcomeres is either or together actin filaments, myosin filaments, in addition to the accessory and regulatory proteins. So what we're going to do now is we're going to look at the structure of each of these separately. Okay, now with actin, these are generally thin filaments that are short. They tend to have a smooth surface and they are greater in number per sarcomere. So if I look at one sarcomere separately and then I count the number of actin filaments that they have or that it has, it's going to be a lot more than the other type of proteins or the other type of filaments. Okay, so if we look at the structure here, this is the structure of one single sarcomere. Okay, you notice that we have a protein in the middle here that's kind of large and irregular, but then it's associated with this small protein here, which is kind of short. It's kind of like threaded or twisted around in this picture. Okay, it has a regular surface, there's no bumps or extensions coming out of it. So this short protein is the actin. Okay? Now, when we compare actin with myosin, just basically think that myosin is going to be everything that's opposite of actin. So myosin is going to be thick, and they're going to be long filaments. Okay? And they tend to have an irregular surface or a rough surface because they have a head, a neck, and a tail. So they basically have extensions coming out of them. And the head is important because it has what we call binding sites. Now, binding sites are sites or specific parts of the head where things can attach to it. Okay? And the structures that attach to the head of the myosin are actin. Okay? Now, if you remember earlier, I said that the way that the filaments are arranged is that they can slide on top of each other. Okay, so if one moves in a certain direction, it'll either pull the other one with it or the other one will end up sliding as well. So obviously for both of these filament types to work together or to act in sync, then they need to be connected together at some point. Okay, so where is this point of attachment? It's at the binding site at the head of the myosin. Okay, and ATP, as you know, is going to act as a source of energy. Again, contraction and change in the structure of the filaments and change in their position is going to be a process that requires energy. So it would make sense that the source of energy is found close to the protein filaments. In general, there are less number of myosin filaments per sarcomere. So remember, I said actin is usually more in number. Okay, so in this picture here, you can see the myosin with its rough, irregular surface, and it's got these extensions coming out of it. Now, each of these is referred to as the myosin head, and you can see that it has like a pink circle here, which is the binding site, and you can also see how the head is attached to the actin filament. So basically, if this ends up sliding or this ends up moving, it will either pull the actin with it, and then the movement will take place. Now, with the accessory and regulatory proteins, what you need to know is that the movement of the filaments needs to take place in sort of like a controlled way or in a controlled environment. So I don't want, for example, one filament to end up sliding a lot more or too fast or sliding in the wrong direction or whatever. So what the accessory regulatory proteins do is that they maintain the alignment of the filaments, so they make sure that they're in the correct position. And they also maintain the speed of filament movement. So again, I don't want one moving too fast or too slow or not moving at all. And they basically control what is going on during a contraction cycle. You're going to get this in more details when you take the physiology of muscle contraction. But generally, just for now, I need you to know that these regulatory proteins basically just make sure that everything is happening in the correct way. Now, each type of filament, whether it's actin or myosin, is associated with a specific type or a specific regulatory protein. The ones that are associated with actin are called troponin and tropomyosin, and the ones that are associated, or the one that's associated with myosin, is called meromyosin. Now, so we've seen how sarcomeres are made up of proteins, and the proteins can either be filaments, actin and myosin, or they can be regulatory proteins. What we're going to look at now is the details of how these proteins are arranged and what they end up looking like in terms of the structure of the sarcomere. Okay, so we're back to our original picture. Okay, remember you've got the thick filament here that has an irregular surface. This is called the myosin, and then you've got the thin filaments that are kind of shorter, excuse me, and these are called the actin filaments, okay? And remember they are arranged parallel to each other, and they are kind of stacked one on top of the other, okay? Now, the way that they're arranged means that in certain places I'm going to find one type of protein, sometimes I'm going to find both, and so forth. So we're just going to look at how that takes place. So in the center here, this entire central part, okay, this is one sarcomere, okay? Now, if you look to the side of the picture here, there's going to be the beginning of the next sarcomere. So remember, multiple sarcomeres give me one muscle cell. Okay, so these are the actin filaments of the neighboring or the next sarcomere. Okay, now the ends of the actin filaments, so these are the actin filaments, are held together by this zigzag disc or this irregular line. Okay, so this one is attached to this one and it's attached here, and then they just kind of anchored or held together in one place. Okay, now this line or this irregular line separates this sarcomere from the next one. Okay, so at the end of each sarcomere, I'm going to have an irregular line that is referred to as the Z line or the Z disc. And what it does is that it separates one full sarcomere from its neighboring sarcomere. Okay? Now, you'll also notice that this region of the sarcomere here consists entirely of actin filaments. Okay? So you've got this empty space here. At the top there's an actin filament, and in the middle here there's an actin filament, but there's no myosin in this region here. Okay? And then if we move on to the next sarcomere here, you're going to find the same thing applies. I've got a region that consists purely of actin filaments. Sorry, wow, I just keep hitting the mouse. I'm sorry. Okay, now, the region of the sarcomere, whether it's one sarcomere or both, okay, that consists purely, oh my dear lord, sorry, that consists purely of actin filaments is referred to as the I band. So the I band is the region of the sarcomere that only has actin or thin filaments. Okay? Now, here in the middle, okay, so if we go past the I band, so at the end of the I band, we're going to notice that this central part here actually consists of a mix. Okay, so I've got both the thin and the thick filaments in this region here. Okay, and I can continue with the thick filament, and then I'm going to find the beginning of another thin filament here. So this central part that's bounded by this green line here consists of a mix of both thick and thin filaments. Okay, now if I have both types of filaments, this is referred to as the A band. So the A band is the region of the sarcomere that has both types of filaments. Okay, now within the A band itself, you're going to find that this central region here consists of what? Only myosin filaments. So this central part here, okay, only has myosin filaments. The actin filaments don't reach this part here. So the part that only has thick filaments is referred to as the H zone. Okay, now within the H zone itself, I'm going to have a set of proteins that cross or connect each myosin filament to the one above it. Okay, so these orange circles here, or this orange line here, okay, this is a line that passes down the center of the H zone, and what it does is that it connects the myosin filaments together. This is called the M line. So basically, depending on what I find in a specific part of the sarcomere, each region is given a specific name. So the Z disc is the thick band that separates the sarcomere from the neighboring one, and it is the anchoring point of the actin filament. So remember, each actin filament was connected to the next one by the Z disc. The H zone, remember, was the region in the center of the sarcomere that had myosin but didn't have actin. So it was purely composed of thick filaments. The M line was the perpendicular line running down the H zone. It was that orange structure in the middle, and it consists of cross-connecting protein filaments. So basically it connects the myosin filaments together. The I band, remember, is the region of the sarcomere where there are only thin filaments, so there was only actin, but there are no superimposing thick filaments, so there was no myosin in the I band. And the A band was the region in the middle that contained a mix of both thick and thin filaments, so it had both actin and myosin. And remember, within the middle of the A band, You had the H zone, which was the region that consisted purely of myosin filaments. Okay, so that wraps up the second part of this lecture, which was just basically a detailed account of the um, microscopic structure of sarcomeres and how they make up the muscle. Okay, we're going to go into the last and final part of this lecture in the next video, so I will see you then.